Okay, so definition of a gene for 2 marks. Now, the only thing I'm not sure about, I should say, is the sequence of bases or the sequence of nucleotide. I'm probably going to say nucleotide bases, therefore. That code for a protein. You can probably say polypeptide as well. Okay, the role of transfer RNA in the production of leptin. Okay, so the ribosome is going to read the messenger RNA and it's going to hold 2 transfer RNA anticodons complementary to the codon. So what are the marks here? Transfer RNA anticodon is complementary to the messenger RNA codon. The ribosome holds 2 transfer RNAs in place at once. I don't know if that's a mark, but probably worth adding. Okay, each transfer RNA carries an amino acid that is specific to its anticodon. So we can say a peptide bond forms between the 2 adjacent amino acids. Adding an amino acid to the polypeptide chain. Okay, let's just self-check this. The transfer RNA anticodon is complementary to the messenger RNA codon. Maybe I should say here it forms hydrogen bonds. A with U and C with G. The ribosome holds 2 transfer RNAs in place at a time. Each transfer RNA carries an amino acid that is specific to its anticodon. Peptide bonds form between the 2 adjacent amino acids, adding an amino acid to the polypeptide chain. Maybe we're saying that the tRNA goes back to the cytoplasm to pick up another amino acid. I don't know why I didn't put that in blue. I was self-checking. I probably should have put it in blue. Okay, describe how the primary structure of leptin enables it to be soluble in water. So we can say that the primary structure of the protein is a sequence of amino acids. What else can we say? We can say that they have a variety of R groups that each have different charges. Now, if this was OCR, I would say that the hydrophobic R groups fold onto the inside of the protein, and the hydrophilic R groups fold onto the outside of the protein, which means the hydrophilic R groups are on the outside, which means it's soluble in water, which have different charges. Let's say some are hydrophilic and some others are hydrophobic. It says specifically the primary structure because the folding is the tertiary structure. I'm a little bit lost. Well, not lost, but I just don't know what it wants. I mean, the primary structure does facilitate it to fold. I don't think it'll be a negative mark. And others are hydrophobic. We can say that the primary structure, the hydrophilic R groups arrange on the outside, making it soluble in water. Maybe one for self-check, that one. Several mutations. Okay, so this is about frame shift mutations involve the insertion or removal of 1 or 2 nucleotides from a gene. Why not 3 is a quick question. No, that's a different question entirely. Describe how a frame shift mutation could result in the production of a leptin with a variety of shorter protein structures. So a frame shift changes all the subsequent codons. Okay, this is happening in the DNA. All the following DNA triplets, and therefore mRNA codons. If a codon is changed from one coding for an amino acid into a stop codon, then translation will be stopped prematurely. And the polypeptide, let's use and a shorter protein will be made. Okay, so a frame shift changes all the following DNA triplets and therefore mRNA codons. That's probably clear enough. If a codon is changed from one coding for an amino acid into a stop codon, maybe just make the word stop a bit clearer, then translation will be stopped prematurely. Maybe I would write. And a shorter protein will be made. It's only 2 marks. I think that's plenty for that. It's all about, obviously, if an amino acid codon gets swapped out for a stop codon, then the ribosome will stop at that point and it won't continue translating. So the primary structure of leptin protein is the sequence of amino acids. Each amino acid has a different R group, which each have different charges. Some are hydrophilic and others are hydrophobic. The hydrophilic R groups arrange themselves on the outside of the protein, making the protein soluble in water. Not sure, not seen it. If hydrophobic and hydrophilic interactions are mentioned here, I don't see any mention of it from memory, in my head that is, in the spec. I will double check, and I have written that lecture for OCR, so I can just add that to the course if that's something that Edexcel are examining here.